Welcome to Leafing Through Science, where we dive into the pages of popular books in the life sciences with the writers who made them happen, from new releases to timeless classics. I'm your host, Hedi Young, and this is part 2 of my conversation with Nicole Rust, author of Elusive Cures, Why Neuroscience Hasn't Solved Brain Disorders and How We Can Change That. If you haven't heard part 1 yet, we recommend starting there, but here's a quick refresher. Nicole explained why treating brain disorders is less like swapping a broken part and more like trying to redirect a hurricane. We looked at the limits of the bench-to-bedside story, why seeing the brain as a domino chain of cause and effect has come up short for treating disorders, and how the brain's very adaptability may also be the source of its fragility. If part 1 set the stage for a whole new way of thinking about the brain, part 2 shows where that shift might take us. From how predicting salmon populations can help predict brain seizures to surprising new insights into why we sleep, Nicole shows how embracing the brain as a complex adaptive system, full of feedback loops and interactions, is opening doors to remarkable discoveries. And along the way, we even make room for a cat quip or two. So settle in as we explore the new grand plan for neuroscience and how this major shift is helping us tackle one of humanity's toughest challenges. Now, there's this great old fable from the 1960s called Chaos in the Brickyard. It compares science to building with bricks, where the bricks are all the facts and findings we collect. At first, the idea was to create exactly the bricks you needed to build something solid. But over time, people started focusing more on making as many bricks as possible and piling them up. The problem was in that huge pile, no one could find the right pieces to actually build anything. It's a reminder that collecting data is important, but without a clear plan for how it all fits together, we can end up with just a big heap instead of a real structure. And a good example of this is how we actually measure brain and mental health. You point out that before we can even start to tackle treatment, we need clear, well agreed upon definitions. For example, having fully standardized ways to measure something like depression.
Yes, absolutely. For a field to progress forward, it's really important that everybody is measuring something like the same thing. In the history of figuring out how to measure temperature, for example, back in the 16th century, we had to create thermometers, and all those thermometers had to say the same thing. So when we do experiments, we can compare across them. By analogy, it's really important that if we want to measure, for example, the outcome of a clinical trial for an antidepressant or a behavioral therapy, we have some way to measure depression and mood and some sort of scale. And one important idea behind that is the idea of standardization. Now, it turns out that today we have a lot of different ways to measure depression. We have over 250 scales for depression, but there are two scales that have been used. Unfortunately, they're used for different types of therapies, so it's really hard to compare between them. So, for pharmaceutical based interventions like antidepressants, there's one particular scale. For behavioral based therapies like a cognitive behavioral or a talk therapy, there's another scale. So, it's very difficult to compare the effectiveness of an antidepressant against a behavioral therapy when the trials are using two different types of measures. In 2019, an international group got together in order to figure out how to standardize depression measurement. And that was terrific because now we're starting to get everyone on the same page about standardization. At the same time, we don't want to get stuck with this one way to measure depression because we're jumping in assuming that we're not going to get this right out of the gate, right? So there is this kind of reciprocal loop. Once we understand what depression is, we'll know exactly how to measure it. But before we understand what depression is, we're taking our best guess. So we have the tension between two things happening. One is the need to standardize things so we can compare the efficacy of different types of interventions or across different studies. But the other is the need to learn and evolve and change. And so that's an ongoing conversation that has to happen in any field, including depression measurement.
And you argue that instead of looking at one factor at a time in isolation, we need to study multiple causes simultaneously.
That is important. So when you're dealing with a complex dynamical system, its holistic properties, its emergent properties, they happen due to interaction between its parts. And so what that means is you have to measure those interactions. It's not rocket science. So imagine that you have a system in which when thing one and thing two are both on, it's active, and otherwise it's inactive. Well, you're not going to figure that out if you just flip on one or two. You have to flip them both on at the same time, right? So this is an example of a nonlinear type of system that has this property. Today, in depression research, there was a kind of a meta-analysis that looked back on depression research over a long swath of the field, over I think 15 years or so. What they found is that 93% of studies of depression, they're really just studying one cause at a time, which might be a genetic cause or insomnia or something else. So it's just an example of how we've really been thinking about the brain more as this domino chain and studying it from that type of perspective, one domino at a time. And what we really need to do is to begin to make these measurements simultaneously. So these happen within the same individual, right? We want to measure all the things. We want to measure their genes. We want to measure brain activity. We want to measure lots of things about individuals as opposed to one thing across many individuals. That's not going to get us there.
Right. So we should be recording activity across different neurons and brain areas all at once, ideally, or measuring genetic variants together within the same individuals.
Yes, absolutely. The interactions we're looking for. Exactly.
So we've talked about feedback loops and how this can challenge our understanding of causality when causes become effects that circle back as causes. And models can really help us to understand how different causes interact to drive disease. Now, people have pointed out issues with models. There's a quip in your book where Arturo Rosenbluth and Norbert Wiener write the best material model of a cat is another cat, preferably the same cat. And there's another famous phrase, all models are wrong, but some are useful. That said, I think it's important for us to acknowledge that models can be very helpful for getting to grips with the interaction of these different causes across different levels. For example, the creators of AlphaFold, who were awarded the Nobel Prize in chemistry in 2024, and they had used a machine learning model to help us understand the principle of protein folding. But interestingly, this was, as far as I understand, Nicole, a black box. So
Mm-hmm.
we weren't really sure how this machine learning model was getting to grips with the problem of protein folding. And incredibly shows that these days you can win a Nobel Prize for cracking a problem without understanding how you cracked it.
Yes, absolutely. Yeah. It's this fascinating time in humanity's history, right? That also has big implications for brain research. It used to be that models were held up as the ultimate test of understanding, right? If you could illustrate that you could build a model and recapitulate whatever phenomenon you have of interest, you really understood it. And in the process of trying to create those models, sometimes scientists would realize they didn't understand as much as they thought they did because they thought that this is how it's worked, but then they built the model and the model doesn't really work. So something must be missing or something must be wrong. For the first time in history, we have this amazing thing called machine learning, right? So we can set up a computer model. It starts all randomly connected. It learns based on feedback from a tutor, and it basically wires itself up figuratively to do whatever needs to be done. And that's how AlphaFold effectively worked. So it took the problem of protein folding is taking genetic sequences and predicting the three-dimensional structure of proteins. And it took lots of examples of how that works, just the mappings. And then it was able to generate, based on that, new predictions for how proteins would fold that we didn't have yet these mappings. There are millions of proteins out in the world, and we had about 100,000 examples so far that have been actually measured. So it can predict the three-dimensional structure for proteins that haven't been measured before, and it does quite a good job at that. What's notable about AlphaFold is it was constructed not based on understanding the principles of protein folding. Like if a positive ion over here is next to a negative ion over here, then that will create a stable configuration, but rather just learning. And we still don't really understand the principles of protein folding, even though AlphaFold exists. So for the first time in history, we have models that work, and we built them, but we don't really understand how they work. And that's really changing the logic of how science works in all of these really fascinating ways. Of course, we're going to use these models, right? Like AlphaFold, it can do a good prediction. This is helping us develop new drugs. Of course, we're going to use them, but it really changes the way that scientists operate, which is fascinating.
And occasionally researchers peek inside these models to help them understand what's going on.
Yes, yes. A great example of that is a problem called cognitive flexibility. So one of the biggest mysteries, I think, in all of brain research is how can we flexibly do different things with the same information. So imagine you're looking at a photograph and you want to look and find yourself, and then you want to look and find someone else. How does that work? How does your brain take that same photograph and do different things with it depending on your goals? And so this is a type of flexibility. It's a hallmark of higher creatures. Not all creatures are incredibly flexible, but the higher you go up on the evolutionary tree, the more flexible we are. So a classic description of how this works is you have this little person inside your head who pulls the lever, right? This is called the Cartesian theater. It's just this idea proposed philosophically by Dan Dennett as a straw man. So obviously that's not how it works. You don't have a little person in your head pulling one lever to look for you and another lever to look for somebody else. But how does it work? That's been a big mystery for decades. And researchers didn't even know how to go about thinking about it, particularly when they were thinking about the brain in this very domino chain way. So there was a big breakthrough circa 2014 or so, where researchers recorded a lot of brain activity data. In this case, it was from monkeys who were performing this task. And it was in a simple world where there were stimuli pictures, they were moving pictures, and they could either move one direction or the other, and they could also have different colors to them. So a very simplified world, reduced to motion and color. But on some trials, the monkey had to determine what direction things were moving. On the other trials, the color. So the question is, well, how is the monkey brain switching between these two things? Where's this little person in the head? The breakthrough was when researchers trained what's called a recurrent neural network. So a neural network in which has this property, if A sends information to B sends information back to A. They trained the recurrent neural network to do the task. They figured out that the recurrent neural network seemed to be doing things like the brain, which when they looked at the brain data, they couldn't make any heads or tails of it. And so they said, well, now we have a network, we can figure out how that's working. And they were able to crack open the box because it's a recurrent neural network and it's just really a bunch of math and computer models in there. They were able to figure out how it works. And it works via these properties. The first property you have to understand is that these recurrent neural networks, they tend to evolve their activity and then get kind of it's drawn dynamically into a decision. So is the motion moving to the right or to the left? That's a point on a line, right? Where you make a decision. If it's over here, I'm going to say right. If it's over here, I'm going to say left. And likewise, if it's one color, I'm going to make this decision. And if another color, I'm going to make this decision. So everything just ended up as a decision about where things fall along a particular line. It's called a line attractor in this field. But then the question is, well, that tells you how the decision is made. But what about the little person pulling the levers? And there they were able to infer that the way the model worked was via what they called a selection vector, which I like to call a shocker signal. So a shocker signal meaning it was just perturbing the neural network, kind of shocking it. If it's a motion trial, you shock it that way so it relaxes to make a decision about motion. And if it's a color trial, you shock it that way so it relaxes to make a decision about color. So for the first time in history, they were able to replace this idea of the little person inside the head pulling levers with an actual proper mathematical model that made us all change how we think about the brain works. So it was the ability to measure something we've never been able to measure before because those researchers shifted away from thinking about the brain as a domino chain like neural network and toward thinking about it as one of these systems that's chock full of these feedback loops. It's a whole different type of math. It's a whole different type of inference, but it's leading us to new discoveries in a really exciting way.
And speaking of one such discovery, an amazing example in the book where you talk about the search for a treatment for GBA1 associated Parkinson's.
Yeah, this is very exciting. So, yeah, so Parkinson's, it's really clear it doesn't have a single cause. It's one of these disorders that has a multitude of underlying causes. For about 5% of individuals with Parkinson's, they have a variant or a mutation in a particular gene called GBA1. And that mutation does not ensure that someone will get Parkinson's, but it increases the risk substantially. And so, for this class of individuals who have GBA1 related Parkinson's, there's been a hunt for trying to come up with a therapy that might work particularly for these individuals. So, GBA1, it turns out to be an enzyme, and it transforms a lipid, let's call it A, into another lipid, let's call that B. So it goes A to B, and there's another enzyme that does the opposite, B to A. And so the first attempt at trying to come up with a drug for GBA1 related Parkinson's just looked at things in a very domino-chainy way and inferred, well, if the enzyme that takes A to B is mutated, maybe it's the accumulation of too much lipid A that's toxic that's causing neurodegeneration. And so they designed a drug around that principle. It didn't work, actually made it worse. So researchers at BL Biotech, they took a step back and they said, we don't think it's as simple as all that. And they mapped out all of these transformations that were happening between lipid A and lipid B, realizing that only a subset of them were happening in this particular organelle that's used to clean up cells called the lysosome. Sometimes things were going outside. And each one of these steps, which were quite long and intricate, they had different dynamics and involved other enzymes as well. So they worked with mathematical modelers in order to map out the entire system as a complex dynamical system and then figure out if this is the problem, GBA1 mutation, how can we shift this system from disease to health? They made a drug based on that principle, and that drug is going through clinical trials. All fingers are crossed, 2026. But it's a great illustration of how the field is shifting away from designing drugs based on kind of simplistic ideas about what's mutated and find the broken domino and fix it, toward much more sophisticated mathematical models where the idea is we want to control the system to shift it from disease to health. And we have to figure out how to do that in a way that's going to require these complex dynamical systems and mathematical models of such systems. It's very exciting.
And so building models can be one way of grappling with this complexity and these interacting causes. But there are also approaches without explicit models, such as this fascinating example you describe of a seizure forecasting clinical trial in Australia.
Yeah, there are these properties of nonlinear complex dynamical systems that make them very special that have been used quite extensively in fields like ecology. And so the seizure forecasting trial you're talking about borrowed these ideas from ecology and applied them to seizure forecasting. So the
It was
need
even
for
used for predicting salmon populations.
Yep, salmon populations, exactly. When the
Right.
red tide will bloom. These are really exciting ideas. And so the issue with seizure forecasting is that when an individual goes into a seizure, it's important that they take medications to mitigate the effects because it can cause brain damage, it can even cause death. So they want to have this medication on board when they're about to go into a seizure, but these medications are sedatives, so you can't have them on board all the time. So it's to great benefit for an individual to know when they're about to have a seizure so they can take this medicine. So the goal is to forecast when a seizure is about to happen. And a clinical trial in Australia, I think it was circa 2010, had individuals implanted into their brains were these electrodes so they can measure brain activity continuously, including when they were having a seizure. And the goal of that clinical trial was to detect when a seizure was going to happen. It wasn't incredibly successful, but what was very successful about that trial was getting the data that we need in order to develop new ways to detect seizures. And so the way that has been most successful to date borrows this idea from ecology. And the idea is that when any system, whether it's an ecosystem or a brain, gets close to a big change, it's called a tipping point, the statistics of that system change. So they become much slower, they become more sluggish, and they tend to move larger. So brain activity will move more slower, sluggishly. So in the brain, they found that this was happening along two time scales. So they could see a rhythm happening in the brain on a day-night rhythm for a lot of people. And they could also see something that was happening at a slower time scale along, it varied from person to person, but along weeks or even months. And when they combined those two statistics together, they came up with the best predictor of seizure that anyone's ever created to date. And it was pretty accurate. It could detect that over 80% of seizures were about to happen with a pretty low false alarm rate. So it didn't falsely predict that they would happen as well. And so, yeah, this is a really great example of how researchers shifting again to thinking about the brain in this new way are able to create and measure things that they've never been able to measure before, borrowing principles from ecology, which is wonderful.
It's incredible how predicting salmon populations can reap understanding in the brain.
Yeah, yeah, absolutely.
We've talked about models and even approaches without explicit models, helping us to tackle the complexity of the brain. But there's also approaches where we have no models at all and we just use a purely behavioral approach. For mental disorders, for example, we have interventions that target the mind, like cognitive behavioral therapy, and this can be effective without any drugs being involved at all. So trying to develop drugs may not always be the best approach depending on the condition.
Yeah, I think that's a very important point. One of the important insights I had when writing this book is just, yeah, rethinking what we want to do in the case of a treatment as engineers think about a control kind of problem. So, in the case of a brain in a disordered state, we want to shift that brain to a healthy state. That's the goal. And that's what engineers would call a control problem, not in the creepy or cartoonish sense of control, mind control, but rather that's really what a treatment is. It's a causal perturbation that shifts the system from one state to the other. And exactly what you need to do to control a system depends a lot on the details of the system. In some cases, there will be some sort of control knob that you can just tweak in order to change things in the way that you want to. But in other cases, that might not work. And in the case of depression, I think we don't really understand yet what depression is, but you could imagine that, and this is one of the leading theories of depression. Depression is a brain that has become entrenched in a maladaptive state. It's kind of learned its way into a disordered state. And what you want to do is you want to figure out how to benevolently reprogram it back to health. What might be required, given how we know how the brain works? The brain has 86 billion neurons in it, right? It's not unreasonable to think, based on what we know about mood and depression, that we might need, in order to shift a brain out of a depressed state, to effectively change how strong millions of neurons are connected together, the ones in the mood network. And there's no particular magic pill that you can take that's going to do that, right? That's going to precisely change all of those strengths of connections, exactly what needs to happen. No holographic kind of laser guided approach. There's no form of brain stimulation that's going to do that. The only way we're really going to do that is the brain has to relearn its way to health. And the way the brain relearns, the most effective way to reprogram those millions of connections is through a talk therapy. So that's working with an individual in order to relearn the way things should be, reconnect all of those brain cells together, or through, as you mentioned, cognitive behavioral therapy, which is really a form of self-talk therapy where you're intervening with your own thoughts and kind of redirecting what's happening in your brain as you kind of control what's going on there. So that's one way to think about what might need to happen for depression. It's not going to be a brain based intervention. That said, it might be the case. Now, we know that those talk therapies they're not effective for everyone. And so the question might be, well, why not? Well, maybe their brains are just so entrenched that they need a little nudge. They need to enhance plasticity. One thing we know is that child brains are much more plastic than adult brains. Adult brains can get stuck, particularly certain parts of the brains. So there is an effort to try to find different drugs that might enhance just plasticity overall to enhance the effectiveness of talk therapy. That might be a really promising path forward.
So we've talked about how models and approaches without explicit models and even just purely behavioral approaches can all address the sheer complexity of the brain. And just thinking of other ways of tackling complexity and finding understanding. You mentioned one example of how research is switching from purely basic research to trying to fix the brain as a way of understanding it. And the example you mentioned is the Stanford Artificial Retina Project.
Yeah. So EJ Chisholensky at Stanford leads this project, and EJ has spent his career trying to tease apart how different types of cells in the eye carry different types of information to the brain. And that was the first phase of his career. And the second phase of his career, he's very motivated to take what he's learned and create a new type of therapy for individuals with blindness that follows from degeneration of what's happening in their eyes. And so this is a form of, it's an electrical stimulation of the eye to carry information to the brain. And what's different about EJ's approach is he tries to use the information about the cell types that he learned in the first part of his career. As a metaphor, you can imagine learning a language, but you won't really understand if you can speak it until you start to interact with someone else, right? Then you could see like if they understand what you're trying to say to them. And so that's what EJ is trying to do is he's trying to understand whether or not the information he thinks he's learned about how the eye works, if he recapitulates that and sends information to the brain, can the brain actually see it? And so that's a wonderful example of someone who is trying to take basic research and then, carry it to the bedside, but also trying to understand in this very causal way that's therapeutically relevant, is the stuff that I learned, does it actually matter for the brain, right? So you can imagine there could be epiphenomenon, correlation does not imply causation. So here we have a causal perturbation that's telling us what really matters for the brain to hear when you try to speak to it about the facts that we've learned. And so this is a wonderful example of a win-win. It's a win for the researchers who are trying to figure out how the brain works, and it's a win on the bedside as well. So those two interacting together, it's a really powerful approach.
Fixing is a way of understanding, yes. But it's also important to recognize just how essential curiosity driven basic research has been in biomedicine. History is full of examples where scientists were studying something with no obvious application and it ended up transforming medicine, like investigations of retroviruses long before HIV was discovered, or research on light sensitive proteins in algae that eventually led to the field of optogenetics, or even chemicals from snails that turned out to help treat pain. And this kind of foundational research is critical for building the knowledge we need to tackle brain disorders. And beyond that, studying the brain simply because we're curious, because we want to understand ourselves and how our minds work, is a meaningful goal in its own right.
Absolutely. I set off to write this book as a foundational researcher, trying to understand the nuts and bolts of how the healthy brain works. On the other side of writing this book, I continue to be a basic or foundational researcher. And that's because I do see that as the bottleneck to society's needs in part. I really do think that we are so limited in trying to even design new therapies because we don't know enough about how the brain works to figure out what's causing the brain disorders in the first place. And so we really have to understand more about how brains work. It's also the case that it is, like you said, it's a worthy end goal in and of itself. We are a curious species, and it's worthy for us to try to understand how we work. Questions like, what is mood? Why does mood exist? This is important not just for individuals with depression, but it's important for all of us. So yes, I think basic or foundational research is as important as the type of research that's directly targeted at therapies. I just don't think we're going to reach society's end goals if we don't do both. We'll be limited.
Yes, important for understanding brains and individual brains too, because something you hear more and more about is personalized medicine. This is an approach that is taken by labs at the Champalimaud Foundation. And the idea is that we need to take into account differences between individuals in order to treat them effectively. In the example of the seizure forecasting study that you described, the goal there is not to create a single fixed algorithm and apply it to all individuals. And that's because the most predictive features are different for different people. For some individuals, the time of day will matter for seizures, but not for others. And for some individuals, all the electrodes will be informative, but for others, they will not be.
Absolutely. Yeah, this problem of individualized medicine, it's such a tricky issue. And it's tricky even, philosophically, you can see how it's tricky. So these disorders, as you said, and it's thought to be true about almost all of them, right? Depression, anxiety, Parkinson's, Alzheimer's disease, they have a multitude of underlying causes due to differences in those individuals. But it creates this really difficult chicken and egg problem. So, how do you even figure out what the cause of something is if you can't group together everybody who has the same cause, right? And differentiate them from individuals who have maybe a different cause or don't have the disorder. So, the agenda is that the way you want to do this is you want to measure a whole bunch of stuff from these individuals, and then you can kind of infer it out by maybe a cluster analysis. Say, okay, well, these individuals with depression, they have this gene, and other individuals, and so on. The issue there is we have to get the measures right. We have to figure out what to measure. And genes and fMRI measures of brain activity to date have not led us to the right measures that allow us to tease all this apart. The dream is that our ability to measure things that we've never been able to measure before, like in the seizure forecasting trial, these new algorithms will lead us to new insights that then will lead us to differentiate the different causes. But we have to get the measures right. And that's why I'm so optimistic about the next steps for brain research, because I see for all of these different functions and dysfunctions, we're finally, for the first time in history, figuring out how to measure them. So I see a lot of hope there.
And on to those next steps, Nicole, because we started the show by talking about the current grand plan, the molecular neuroscience framework, how typically we go from genes and proteins to neurons and circuits to brain functions, dysfunctions, and the environment, with there being one feedback loop from the environment. But now you propose a framework where we think of the brain as a complex adaptive system with reciprocal connections between all of those things.
Yes. So the agenda of the 1990s, we thought about the brain as a domino chain, right? Causes lead to effects. And you could think about the overarching plan for brain research as figure out which dominoes in the chain are broken and then go in and target them for effects. Now, when we think about incorporating all of these feedback loops, the plan changes. And the question is, how do we even describe what the plan is, right? We're dealing with something that's much more complex. And I think that the plan has two components to it. One, it's a deeper appreciation that we really do need to build mathematical computational models because we can't intuit our way through these systems. They're just too complicated to try to tease apart how they work. And two, we really have to begin to think about this as a benevolent type of control. So given the models, we have to look at the models and ask, how can we take this system when it's in an unhealthy state and shift it back to health? So I would describe the new plan as model these systems as complex adaptive systems and figure out how to shift them from disease to health. That's kind of the new plan that I think is emerging of this era. And I want to just make sure it's very clear, this isn't my genius idea. When I set out to write this book, I didn't set out as like Nicole's going to swoop in here and figure all this out. For me, it was really, I think about myself as an author, channeling the genius of this era and taking the genius from all different areas of brain research and try to infer what is the genius of this era. And I really do think that that's when we fast forward in 30 years and look back and ask, what was the genius of the 2020s? That's what we're going to point to. We're going to point to this shift in thinking about the brain, not in the 1990s way, but embracing its complexity for societal good.
So now, instead of find a mutated gene, make a mouse, create a drug, we have create and test models of the brain as a complex adaptive system to infer fragility and determine control.
Absolutely. You nailed it. That's the new plan.
And there's no magic bullet, because it's likely that for all these different disorders that we've been talking about, there are going to be different solutions. And even within each of those disorders, as we were discussing with the variation across individuals, there will be different solutions for different individuals.
Yes, yes. I think that that's a very important component of that. Just because we solve paralysis does not mean we've solved depression. For example, the motor system and the mood system, they have different constraints. They're there for different purposes. We're going to have to take each one of these systems and their functions and their dysfunctions one by one and attack each one of them independently. That said, there will be some frameworks that we can carry across these different systems, but we have to be very careful about not overgeneralizing across them.
Right, because for Parkinson's or Alzheimer's disease, for example, the goal there is to shift cells from degenerating back to
Exactly.
healthy functioning. In the case of
Mhm.
depression, we need to develop a model of mood. In the case
Yeah,
of Parkinson's, we create a model of the factors that support healthy dopamine neurons.
absolutely. Yeah, so important.
And give a metaphor for thinking of the brain as a two-sided coin
Mm.
poised at the knife edge, and how that can help our understanding, even give us new insights into why we sleep, for example.
Yeah. So as I think about the shift away from the domino chain and think about, the new metaphors we need to use for thinking about the brain, I've converged on thinking about the brain, yes, as a two-sided coin. And on one side, we have a description of the brain's functions, things like seeing and remembering and moving. And on the other side, we have the need for the brain to keep itself in healthy balance. The way these two are related is that once we shift to thinking about the brain as a system with these feedback loops, so if A sends information to B, B sends information back to A again, we know that systems like that really do need to keep themselves in balance. So if you have just excitation in a feedback loop, that's an amplifier, and a brain like that will quickly enter a seizure, which is trivial in a computer model. It's very easy to make a model of a seizure. It's actually hard to make it not seize when you have these big feedback loops. So how does the brain achieve it? The brain does that by keeping balanced excitation and inhibition, positives and negatives, in this perfect balance. But the problem is if you have too many of the positives, you do end up in a seizure. But also, if you have too much of the negative, you end up with a brain that's in a coma. What you really need to do to make a brain work that has these reciprocal loops is to keep these things in this exquisite balance. This balance point has a lot of really interesting properties. And in physics, you can also see systems like this. It's a state called criticality. And at that point where the brain is perfectly balanced, it can process the incoming sensory information in the optimal way. So it's a really special part of the brain. So thinking about the brain in this way is leading researchers to new insights into how the brain functions, new ability to measure things we've never been able to measure before, and insights into things like why do our brains need to sleep, with the idea that sleep exists to rebalance the brain. So the evidence for that comes from Keith Hengen and his team at Washington University in St. Louis. So they're doing these very clever experiments. In this case, they're in rats. So what they're doing is they're measuring from the rats' brains 24 hours a day, where the rats are doing all their rat-like things. Sometimes they're eating, sometimes they're playing with their cage mates, and sometimes they're sleeping. And what they're finding is that in the beginning of the day, the rats' brains, when they wake up, they're right at that balance point. As the day progresses, their brains become unbalanced, but sleep brings them back to that balance point again. And to make sure that it's not just a day night rhythm, one of the things they did is they took the rats at the end of the day, they kept them up 90 minutes beyond their bedtime, which they saw their brains became even more imbalanced, suggesting it's not just circadian. And again, sleep then brings them back to this balance point. So this is really exciting because it's giving us insight into why is it that our brains need to sleep? They need to do it to rebalance themselves. And it's also giving us new ways to measure things that we've never been able to measure before, like how sleep restored a brain is. And this is important because one of the hypotheses of Alzheimer's disease and what's happening during Alzheimer's is that maybe this is a brain that can't sleep restore itself. For the first time, we're starting to create new ways we can actually measure how sleep restored a brain is, which then could lead us to test hypotheses like that we've never been able to test before. So really exciting example of the two-sided coin, right? A brain that to do its functions, it has to keep itself in balance. And the way it does that is by somehow renormalizing itself during sleep.
Right. So homeostasis, keeping a
Mm-hmm.
balance, isn't just applicable to the body, it's also applicable to the brain.
Yes, wisdom of the body, wisdom of the brain. Yeah.
And how we are always at a knife edge between order and chaos.
Mm-hmm. Yes, yes, that's an important theory.
And so there are also many other impediments to new therapies, which you briefly mention in the book, each of which is probably a book in itself. Um,
Absolutely.
for example, there's the clinical trial phase, which is
Hmm.
both incredibly expensive and incredibly risky. On average, it costs about $1 billion and takes around 10 years to test just one brain drug. And even
Yeah.
then, fewer than 5% of those drugs actually make it all the way to the market.
Yes, yeah, there are all sorts of things I think that are impeding progress. And so I don't want to make it sound that, oh, if we just shift to this new way of thinking about the brain, everything is going to be fixed. I decided to write this book around the issue that I didn't see was getting enough attention, where some of these other issues have gotten a lot of attention. And there are a lot of great minds trying to think through how to fix problems like science that doesn't reproduce when somebody else tries to do it, or the incentive structures for scientists, to do really novel things. So lots of great minds trying to think about how to get scientists to do those sorts of things. And Elusive Cures, I wanted to point to one that goes all the way back to the earliest stages of how do we even figure out the brain's functions, not to mention its dysfunctions, and really kind of raise awareness around the need to shift in this way.
Right. And before the clinical trials even begin, there's also the brain research phase. And so that's another aspect of neuroscience which one can look at. For example, there are different ways of making discoveries. You can do
Hmm.
it in a lab with 10 people, a principal investigator, a sprinkling of postdocs and
Hmm.
PhD students and technicians. And then at the other extreme, you have places like the Allen Institute, where you have armies doing experiments together in a very
Hmm.
different setup.
Yeah. Yeah, absolutely. I think that what you've just pointed to is sometimes called big team science, right? And then the alternative might be little team science. And when the optimal thing happens is often about when a field is ready for it, right? So, for example, we know a lot about the visual system, and the Allen Institute is helping us like really move the needle on understanding how the visual system is working. We know a little bit less about, say, the mood system. And so, having a whole bunch of people investing all of their eggs in a particular basket for a particular experiment, right? It might be a little bit too early for that because we just don't even know what experiment should be done. So, there it helps to have a lot of different teams working on a lot of different things to kind of explore the bigger space. So, it's a very tricky system for the community as a whole to navigate, but I think they're doing a pretty good job overall with that balance.
And we have a lot of cancer research, not just
Hmm.
neuroscience research being done at the Champalamo Foundation.
Hmm.
And presumably, much of what we've been talking about also applies to cancer research.
Yeah, for sure. So I think the cancer researchers were ahead of the game in thinking about big genetic networks that they're dealing with as these complex dynamical systems that they need to really understand kind of holistically in order to figure out how to block cancer from happening. And there's some great examples of that. For example, in Elusive Cures, I talk about an example of a form of leukemia which, like all forms of cancer, it's a system that has figured out how to shut off its normal cell death. And so you have to figure out, well, how do we turn that back on again so the cells don't overproliferate and they actually do die as they should. And in that case, they created these wonderful elaborate models of these 60 different genes and their proteins and how they interact. And if you look at this, there's no way to infer how to control the system. You just can't. It's just way too complicated. But by building a complex dynamical systems model, they could then go through this process known as model reduction, where they could say, well, we know that the system is doing a whole bunch of things other than regulating cell death. It's doing all sorts of things the cell needs. So let's get rid of all of the gene proteins that we don't need in order to describe that. And that left them with just 5 genes. And then they could figure out, oh, we understand exactly how to switch this thing on and off. So it's a beautiful example of modeling a complex dynamical system and figuring out how to control it by building these elaborate models and then backing them off and making them simple enough in order to figure out how to develop therapies.
And just before we finish, Nicole, I just want to briefly talk about something you bring up in the book, which is stepping back, looking at things as processes,
Hmm.
not just things. That basically everything is constantly changing. And I think this is a quite a powerful idea because it applies not just to how we do science and how we we look at problems in science and the brain, but also it's just generally in life. I mean, we
Mm-hmm.
as individuals are always in flux. I mean, I'm not the same person that I was 10 years ago, and neither are you, I suspect.
No. Yeah, there's some sameness and there's some differentness, right? And so, yeah, so the distinction between things versus processes, in some senses, a noun verb distinction. So there's you, there's me, there's a cat, a pencil. When we talk about things as nouns, we're really focusing on their permanence in some senses. Whereas when we talk about things as verbs, we really are kind of talking about it as the changing nature of it. So it's raining. And we might want to use the example of, say, a cat, right? So cats are not immortal, but when we call them a cat, we're really focusing on the permanent nature of cat as opposed to calling it catting, right? So the cat is an impermanent thing. So in some senses, this seems a little bit philosophical. But in brain research, it's really important, I think, to think about, are there parts of the brain that we're misunderstanding because we're assuming they're things and they're actually processes? One example I would hold up is the visual system as something that we really do think of as a thing. So a mature adult visual system is often thought of as an information processing machine. Its job is to take information that lands on the eye and do things like label this thing right here as a cup, right? So it's really good at its job, and it's learned to do that across development. So the visual system is really thought of as a thing. And it did start as a process, but the process was more the childlike bits that was learning and dynamically changing. There are other systems in the brain, and I would say mood is one we've been talking about, where it just doesn't make sense to think about them as a thing because their whole point is to continuously learn and adapt to change. If you turn off that learning and adaptation, mood makes no sense, right? It's definitely a process. It's not a static thing that's there to just like map, inputs to outputs. And so mood is definitely a process. An adult visual system might be more like a thing. But yeah, thinking about, well, how much of the brain have we been misunderstanding because we've been making it thing-like, but it's actually process-like. And it's unquestionably the thing-like parts of the brain that we understand much better than the process-like parts of the brain. Those are still very, very mysterious to us.
And not just misunderstanding the brain, perhaps, by seeing it as a thing rather than a process, but misunderstanding ourselves.
Mm-hmm.
We may be more accurate to think of ourselves as processes, as constantly evolving beings, rather than fixed objects.
Absolutely.
So
Yeah.
instead of I think, therefore I am thinking, therefore I'm amming.
I like that. Well said.
Well, I think that brings us to the end of our conversation. I really enjoyed the book and I strongly recommend people read it. It's beautifully written and, in my view, essential reading, not just for people working in the field, but for anyone interested in how science evolves. And I think too often we don't talk enough about this kind of big picture perspective neuroscience. And I think it can help make research more efficient and effective and help us to build more real edifices rather than piles of bricks.
Thank you so much for that, Hedi. I really, really appreciate it. Thank you.
We always end the show, Nicole, by asking guests if they would like to recommend a book, fiction or nonfiction, to our listeners.
Yeah, I want to cheat and I want to offer up two for different swaths of your listenership. The first would be a book that really was transformative for me. It's written by Hasuk Chang, who's a philosopher. It's called Inventing Temperature. And I'm always struck by how many brain researchers have not heard of this book, given how many brain researchers, every time they read it, it transforms them. So I'm not the only one. And that book goes through how is it that we traversed from a sense of hot and cold to theories, sophisticated theories about how temperature works over about 250 years. So it's a fascinating case study. And how do we go about understanding anything? So I recommend that to anybody who wants to do kind of a deeper dive. And I think the other book I would recommend for somebody who's maybe more interested in understanding the humanity of these conditions would be a book by Esme Weijin Wang called The Collected Schizophrenias, which is her first-hand account of what it's like to live with schizophrenia and psychosis, and written in the most beautiful and profound way. And it would definitely, just to understand what those individuals are going through, I think it's one of the best sources of that for anyone who's curious.
Fantastic. Nicole, thank you so much. And to everyone listening, thanks for joining us and see you next time.
Thank you so much. I appreciate it.